So it's my pleasure to be here this morning. It is morning. I'm just flew in last night, so I'm very pleased to be here. First of all, I'm doing my official conflict of interest disclosure. I have no relevant financial information or influence or organization, but I do have affiliation with UC Davis. As Eddie mentioned, I'm associate dean, and I'm also a professor in animal science, and I'm on the board of directors for OFA. So with that, I'd like to go into the actual talk. What you heard earlier were discussions of how to get the testing and things like that into your practice. And this talk is essentially about essentials. What is essential to know about testing and what is essential to interpret them. And so many of the tests will reveal things that are underlying the animal, and you need to know about them. Health screening in general is important for all dogs. We're not just talking purebred dogs, we're talking all dogs, because it can inform and provide health information that allows you to develop long-term, short-term, intermediate health care programs for every dog. In addition, we usually think about genetic screening important for purebred dog breeders to reduce the incidence of genetically inherited conditions. But we need to think about health screening in a broader sense. In addition, AVMA puts a lot of emphasis on health screening because this is a recent policy that AVMA just approved where maximizing the health and welfare of companion animals, and that requires educating the profession and breeders and the public. Oops. I'm very professional up here. I apologize. The AVMA encourages veterinarians to educate. So AVMA also considers health screening very, very essential. So what are some of the uses? Well, radiography, you can use that to reveal joint conditions. OFA started, as you heard in the previous talk, for hip dysplasia and looking at orthopedic conditions. And this is an example. You see an OCD lesion, you can coordinate exercise, whether you need nutrition, what kind of intervention or perhaps surgery is necessary. You can assist the owner in establishing that program for the animal. Another example of use, some herding breeds, and that includes breeds that are not necessarily your immediate idea they may be a herding breed, as well as mixed breed or random bred dogs that have herding heritage, have a genetic mutation that creates susceptibility to life-threatening conditions. The MDR1 multi-drug resistant gene test. If that dog is affected and has the mutation, the brain cannot process the drugs, whether it is an antibiotic or sedative, et cetera, and that can cause a lethality or really impair their health. Knowing that for any dog is really useful. And so it allows you to provide the best care. So what is essential to know about tests? Are they valid? Everybody likes tests. Well, not if you're a student. But are the tests valid? Are they relevant? Are they relevant to this breed, this dog? There's more tests available and we need to know whether they're relevant. And then how is that information used? Can it be used in an appropriate way to improve the health of the animal? So let's talk about validity because that's one of the key pieces. So does the test actually assess what the test was designed to test? And that reflects who designed the protocols for the test and how was that test designed. So if you're doing radiographic assessment, you want the team of the premier radiologists developing the test and the criteria to apply to the results of that test. If you're looking at a genetic test, you want to make certain that the people doing the genetics and have done the experiments know and have targeted the actual thing you're trying to determine. And then importantly, is the test reproducible? So are the values, the test results that you obtain in Florida the same as you obtain in California? And is it the same that you would obtain in South America, Europe, across the globe? Because some tests may have been designed for certain populations of dogs, and that result may hold in that population, but not necessarily broadly applicable. So you need to make sure that the test that's being employed is appropriate. So test validity and trustworthiness. We talk about accuracy and precision in any kind of test. Accuracy is how close a measurement is to the gold standard, what's real. So if my arrow here, so let's say this is the reference value, and our test gives values over here. The accuracy is a bit off from the reference value, but if the measurement is consistently off so that you can make an adjustment for it, you're all right. And then precision is how closely those values are in terms of reproducibility to that the gold standard. So if you look at the bullseye, that might be a nice way to imagine. The bullseye in the center is really your gold standard, what you're trying to hit. And so these target are sort of near the middle, but they're rather dispersed. So that would be the accuracy. And then the precision is how closely the values you get multiple times are to one another. So those two factors go into validity of a test. So what could confound or negatively influence testing validity? Well, it could be the type of test. And if we talk about hormones or measuring antibodies for something, the antibodies have to be present. If you test too soon, those antibodies won't be present to an infection or autoantibodies, and so you're going to get false results. Alternatively, if there are autoantibodies to some tissue, and that tissue is destroyed, over time those autoantibodies will no longer be necessary and they will be gone. So if you test too late, you're not going to get appropriate or accurate results either. We also need to think about cross-reactivity of antibodies if you're using an antibody for hormone or any of those peptide tests. If the antibody is promiscuous, it's going to recognize many other components in your tissue sample or your serum sample and give you erroneous results. If it's a DNA test and you're looking at DNA primers, that has to target the appropriate region of the DNA. And if those primers are not as directed to that region of DNA, then you're going to get erroneous or mixed results. Then we also have to think about confounding factors that are related to the physiological status. Earlier this morning, you heard about doing OFA hip assessment. And they asked the question, what age can you evaluate the OFA hips? If you do it too early, I have a tiny little litter of pups at home right now. They're 4 weeks old. To evaluate their hips now would be absurd because they're in the growth stage. So you have to really look at what time frame and physiological status for the accuracy of the test. In any test, you have to balance cost effectiveness versus the speed of results versus the accuracy. There are tests that are going to be phenomenally accurate, but if they're so expensive no one uses them, that's not very helpful to you. Then test results may be binary. They may be yes or no. Yes, you have it. no, you don't. That's pretty easy to interpret. But some of the test results may be relational. It's bigger or smaller, or it's outside of the normal range. And then when we say it's low normal, we're all overachievers. Well, low normal, I want it higher. I want to be high normal. I want to be really good. And so low normal may be just fine, but people may need assistance in interpreting that. We also have to know whether the test features, the results are predictive. Do they account for percentage risk? And that is a difficult concept for people to get their minds around. Is it high risk, low risk? Well, what risk is bad risk? And what is the proportion of the test? If the test may only account for a certain percentage, because many of the traits are complex. So maybe that test only accounts for a small percentage of the expression of that trait. So then you think, well, what do I do with all this information? And that's where clinicians can help to interpret those results and put it into context for the client. The other piece to think about is sometimes the results, even if it's binary, yes or no, negative or positive, positive may be good in one case and not so good in the other. So it's positive for normalcy or positive for abnormalcy, and the clients need to know that. So the two big buckets of health screening or health testing, we can talk about health screening in terms of physical examination of an individual for normalcy. You're looking at their phenotype. You're looking at what the animal looks like. The other bucket or bin of health testing is genetic testing. And in that case, you're looking at the DNA, and the DNA for propensity for that animal to express the condition, that animal to potentially pass on the condition. And that's where we think about the DNA either as a small mutation or large mutation. So when you do physical screening, as I mentioned, you're looking at the individual, the phenotype, what the animal is expressing. And a health screen can disclose an infectious agent. I mentioned antibody testing. It may be an inherited condition that the animal itself is showing you. Or it may disclose something that's not inherited, but it's a condition nonetheless, and it's a developmental aberration that happened in utero, et cetera. And you're talking about health screening in a diagnostic test. So the types of health screening: physical manipulation. You can have patellar luxation and you can move the patella. It can be visual appraisal where you need special apparatus, so different ocular exams, or the bear testing for hearing. That's a little puppy with the bear testing. Or you can have auscultation or cardiac. You can also look at blood parameters and body fluids. So if you're looking for hormones, for example, thyroid levels or adrenal function, hypoadrenal corticism or hyperadrenal corticism, you would look at the adrenal gland function in response to exogenous administration of a hormone and seeing how it responds, or metabolites. And with these kind of blood parameters or body fluids, you have to have proper sample collection. If you're doing a urine analysis, you want to get clear urine and not contamination. You need to have the right blood sample if you're doing blood analysis and using the right tube to collect the blood, because that can be a confounding effect. Then there's radiographic imaging, and the next talk is going to be presenting proper positioning, because you have to have proper positioning to get that accuracy and precision. This is an illustration of the same dog with different position. So you can see that the femurs are positioned completely differently in the two, and that's going to change how the hip sits in the socket. And so your reproducibility depends upon that, and your accuracy of your test depends upon how that positioning occurred. And then you can do, for health screening, biopsies, skin punch, bone marrow, intestine, you need to make sure you get the right location. If you are doing a biopsy for tumor and you miss, you may give the wrong impression to your client. The other bin is genetic testing. And that is an area that I do most of my research in. And genetic testing is targeting the DNA. And what that does is also rely upon specific laboratory tests that you have with a health screening. You're using either physical manipulation or you're doing some sort of collection of tissue and you're sending it off to a laboratory. But with the DNA, you're really looking and having to send that off to a specific laboratory. And the genetic testing is looking at alterations in the genes that underlie specific traits. And that DNA can describe, we call them DNA signatures. It might be a single point change, but those signatures represent particular breeds. It could represent particular health conditions. It can represent particular individual dogs. There's lots of different ways that we can utilize the genetics signatures. I mentioned parentage, and I'll go into that in a little bit later, and it's also can look at genetic diversity. So the genetic testing, I mentioned earlier the MDR1, the multi-drug resistance gene. And when you get a genetic test for that, you would know whether the dog was affected with that mutation or whether it wasn't. And if the dog was affected, then you would design a treatment plan for that particular dog. The genetic testing can also provide owners with the knowledge of a future condition. So sometimes conditions take time to express, and if you have a genetic test for that, you would know whether that dog would, in the future, express that condition. And so you can have the ability to develop short-term or immediate health plans, the intermediate health plan, and then the longer term. And so that, again, just to reemphasize, that's not just for purebreds, that's for any dog, because that will give you and your owner and the dog a bigger, broader picture of their health condition. So genetic testing, I mentioned it can be important for an individual dog, whether it is a purebred or a random bred, but we can also utilize the genetic testing to assist breeders in passing on best traits to the next generation. So this little cartoon just shows you, well, if you have clear dogs, you would presume 100% clear progeny. If you had a clear and a carrier, your standard, this is for basic Mendel single trait. This is the easy stuff. That's why we can show it that way. You would get the proportional expectation. And then we also need to know that the traits that we're testing for in genetics are not just health traits. There are particular traits that may be relevant to breeders respective of coat color, et cetera. I put more than 150 available genetic tests because they are coming online so quickly we can't keep track, well, I can't keep track of them on all of them, but they are being developed as we are sitting here. People are developing their additional genetic testing. But knowing that you have a clear dog and affected dog, when bred together, give you 100% carriers. If it's a trait that requires 2 mutations to express, then these dogs are not going to express, but they could pass on to the next generation. And that's where that knowledge is helpful. DNA signatures. I mentioned that when we look at MDR1, that's a signature because it's unique to that particular dog, but we can also look at a broader range, and the signatures distinguish particular sequence characteristic of particular breeds. So some of the DNA testing that are for the mixed breed population, people want to know what their dogs are. That would be this using DNA signatures that are reflective of particular breeds. In addition, you can have individual variability in sequences of a particular dog. My DNA is going to be unique to me. It's different than Eddie's DNA. We're both human, but our DNAs are unique, and that gives that signature. And those unique identifiers for a particular dog are really valuable in parentage testing because Fran mentioned there were some accidental breedings that happen. Sometimes people have more than one animal and they may breed and you don't necessarily know who's the father. The phrase I'm sure you've all heard is mama's baby, daddy's maybe. Forensics. Forensics is an ability to identify a particular dog as being distinct. So many homeowners, HOAs, homeowner associations in condos or apartments, they'll allow dogs, but you have to validate that you're picking up after the dog. They'll do a DNA test for each of the dogs in the apartment complex, and then if there is dog fecal matter that is found, they'll DNA test the dog fecal matter, and then link it to who didn't pick up, and then they'll find those people. Alternatively, it's used in forensics for identifying this was a colleague of mine. I have to tell this story because it's fun. A colleague of mine was assessing the probability of a DNA signature of a dog that they linked a suspect to a particular crime scene because of the dog fecal matter on the suspect's shoe, and it was unique to that dog at the crime scene. So it can be used in forensics. DNA parentage, this is just to show you, if you have ever seen that, the sire, they use multiple areas to get that signature. There's the sire, there's the dam, and then you can see that the offspring has a likelihood of being from that particular sire and dam combination. So you've got the double green on the sire, the green on the dam, and then you get the offspring. So it's a way of confirming. Now, genetic testing, I mentioned before, is analyzed by special laboratories. Those are the region or the labs that have the ability to look at the DNA level. Importantly, it's commercially available. I say importantly because you as the clinician don't have to have the test run. Your client, the general public, can run the test, send their sample off to have the test run themselves. So in a health screening, you as a veterinarian are doing the blood draw. You're seeing the animal. You're doing the manipulations, the palpations. In some of these genetic tests, the owner can go get, some of the tests are using the little go-between brushes that you get in a dental office, rubbing the inside of their dog's mouth, sending it off and getting results. Then they run to you as the veterinarian and say, what does this mean? So you are in a wonderful position to be able to help interpret those results. Now, some of the genetic testing are individual tests, so you can get tests for MDR1 itself, or they're composite tests, depending upon particular breeds. Some breeds have multiple tests that they would like run. For instance, Jerry mentioned the Portuguese water dogs are very, very dedicated to health, and they have a lot of health genetic testing that they ask for in addition to other screening. So you can get panels of genetic tests for those different dogs, or there are even larger composite tests that will run many, many different tests on a given DNA sample. Some of those tests are appropriate for a breed, but not broadly applicable. So when an owner sends off and gets 150 test results, and only 4 are relevant to their breed, let's say, that's an opportunity for you to help them understand what's applicable and what's not for their particular dog. So getting the DNA for analyses. There's different ways where you need to get DNA just for DNA amplification so that you would have an ability to look at a particular gene. And that would do, you can collect off of swabs. These are mouth buccal swabs. These are saliva paddles where it collects the saliva and your DNA can be collected there. Or you can collect in blood tubes. These are EDTA tubes, which is really the preferred collection for your DNA for analyses. Some tests will look at chromosome structure, which is a karyotype, looking at the actual chromosomes, and they require specific blood draws for that. So your owners are not going to be able to do karyotype testing. So the types of genetic tests, there are direct mutation-based, and that's important to understand that there are different quality levels of DNA testing. So the direct or mutation-based is looking at the actual alteration of the gene's DNA for that gene. So here's a normal, here's an A in this location, and in a mutant, the C. And so and that change from an A to a C would be responsible for the condition. So direct tests, when we look at DNA differences, I mentioned that my DNA signature is different than Eddie's. It's different than all the rest of you in this room, too. Fortunately for everybody, I'm not a twin, so I'm unique to everybody else as well. So DNA is going to be different. That's normal. But different DNA does not mean that the difference is going to change a gene product. Because there are changes in DNA that we say are neutral. They're not really causing a difference because of the way the DNA works in converting to proteins. But causal mutations or DNA can result in changes in the actual protein. So we can talk about nonsense mutations. In this case, the C is changed to a T, and that introduces what we call a stop codon, and that causes the protein to stop and be short. And so that changes the actual protein. That mutation of that C to a T changes the actual function of the protein. So that is a direct mutation that is causal, and it changes it in a shortening. It can also be a missense, where in this case the A is changed to a C, and instead of a histidine protein or amino acid, we put in a proline. That proline can change the form of the protein, and that protein will no longer function in the same fashion. Remember I said some mutations may be mutations but don't change anything? If that A was changed to something that didn't change the proline, or didn't change the histidine to a proline, or change the histidine at all, that would be a difference, but not affecting a change. Does that kind of make sense to people? Okay. In addition, a direct test can assess whether there was an insertion. So an additional DNA base may be added so that between the T and the C here, an A was added. And that shifts the whole sequence. So the code is different, and it changes the amino acids that come in, and that changes the actual protein structure. Again, causal mutation. In addition, or in editing, that A may be lost, and that too will change the amino acids and change the protein. These direct tests are targeting that region that is changed. We can also have mutations where we get an expansion. So in this case, this CAG region right here, there's an error in copying the DNA, and CAG is repeated, and CAG is repeated, and so instead of just one glycine here, we have 3 of them, and that changes glutamine, that changes the protein that comes from that DNA sequence. And direct tests, and I'm emphasizing this very explicitly because the direct test is targeting that particular section of the DNA, and we know that that section of DNA, when it's changed, changes the protein, which changes the way the condition is expressed. That duplication I just mentioned can be really extensive. So in this case, this region of the chromosome was completely repeated. And so now you have twice as much than you would normally have, and sometimes twice as much is not good. More is not better. And in this case, this is a situation in canine cancers where you have trisomy, three of the chromosome 13s instead of 2, and that, again, a little is good, too much is too much. So the direct goal of a direct test is to look at that particular region of DNA. To do it, I mentioned we've got those brushes, or you can get the blood, and basically what happens in the laboratory is they extract the cells from that blood or from that buccal sample to isolate the chromosomes from the nucleus of the cell, and then you expand it, and then you have one copy. One copy is not enough, so you amplify it with what's known as polymerase chain reaction. It's just an amplification. You take one and you make a whole bunch of copies, and once you have a whole bunch of those copies of that region, so that direct test region, let's say right in here, we've made a whole bunch of copies. I'm probably moving that arrow around way too fast and you're getting seasick. You make a lot of copies so that it allows you to actually detect the sequence of the DNA. Then you run it on different arrays, and when it is a C or a G, you can tell because of the way there's a tag on it. So in this case, we have the appropriate tag coming in, matching that A and a T, and there you have it. You have the ability to say, this dog has a T, this dog has a G, and that's just the way it's assessed in the laboratory. So if the direct test targets the actual mutation, and that causes the condition, it should be 100% accurate, right? Well, what could possibly go wrong? Lots of things can go wrong, especially if a cat jumps in the water. My cats don't like water. What influences 100% accuracy? Well, the quality of the test. The test needs to target the proper DNA. Remember early on I said, Who developed the test? How specific? How accurate? How precise is it? And if it relies upon sequence data, remember that DNA is all those A's, G's, and T's, and it has to be in a particular order. It needs to be accurate in sequencing. And so here is the DNA sequence that comes off of a column. More than you ever want to know, I'm sure. Well, maybe not. But this is pretty straightforward. You have some nice peaks, and each peak will give you, with a different color, the particular DNA sequence. However, that's perfection. Down here, not so ideal. Can you read that? Could you really see those tight peaks illustrate a particular base? In this case, that peak is a T. It'd be hard to tell if that is a peak or not because of all the background. Or here, that's just a mess, as is this. Why am I showing you this? Because that happens. When the DNA quality is not good, you get junk. So if you have a blood sample that's collected and it doesn't get sent off for a while, do you think that the DNA in those cells are going to be, it's going to be maintained over time? It's going to degrade. That's the way DNA does. It degrades, and then you get these ugly sequences. So that can influence how accurately you can detect whether a mutation is present or not. The quality of the lab can influence the results. The contamination, maybe the laboratory is running multiple species at the same time, or they're mixing up the samples, or think about the owner who sent in the sample. The owner decides they're going to sample 5 dogs, and this dog just ate a bunch of kibble. They sample the inside of the mouth, and there's tons of kibble on that instead of actual DNA from the cell of the mouth. Or they've got the 5 dogs and wasn't that Fluffy? I'm not sure. Is that Fluffy? I don't know. We'll write Fluffy down on the name. So you've got this problem with submission. You've got accuracy and precision of the tests. That is essential for validity of a genetic test. So it has to be accurate and it has to be precise. And if the results are off, you might want to redo it to confirm the test. You would always get a second opinion if you were diagnosed with a condition yourself, wouldn't you? Before you started doing all these treatments, that would be a great thing to do, just to make sure with the accuracy and precision. And then again, there's standards of the tests that are used. You need to understand and appreciate that sometimes there needs to be validation, that when we do this amplification of the DNA, make sure that there's not contamination floating around in the laboratory to give false results. Some mutations hold for a given subpopulation of a breed, but not necessarily individuals of the breed. What's the science that supports the test? If you go back to one of the previous slides I talked about, you have research that establishes a test in a population. That population of dogs, let's say you were working with a closed population of, well, I'll give you an example. I work with a closed population of a service dog breeding company or organization, enterprise. If I found a mutation and a test that was appropriate for that population, am I done? No, because that population may be unique. Now I would need to test it in this population over here and in this population over there to make sure that it's reproducible and applicable across the globe, not just in someone's personal breeding colony. And so that's the science that supports. Some tests are breed specific. So DM, or degenerative myelopathy, there are tests for that, and 2 copies of the SOD1, superoxide dismutase 1, in case you wondered, confer increased risk for DM in some, but not all breeds of dogs. And even dogs that are mutant two copies, so they have a mutation DM, they may not necessarily develop the disease. So we know that there is variable presentation of the disease between breeds, which would suggest that there are other factors that influence expression, not just genetics. And we also wonder, well, maybe we know DM is late onset, so maybe dogs that are homozygous mutation for DM, in those breeds where it doesn't show up, maybe it's because they're not living long enough or there's some other extenuating. We don't know, but that's something to think about when you get those results. Especially in a breed that DM has not been associated with, and especially when you're getting a panel where it says, well, this breed here has no DM, people are like excited, or it has DM, but it's never been shown to express in that breed. You just have to be aware of that. Another example is hemophilia A. Different mutations in the same gene cause the disease in different breeds. So if you use the wrong test on one breed, you'll get a false sense of security or incorrect conclusion. Let's say you're using the Boxer test on a German Shepherd. That's not going to be appropriate. So why I'm emphasizing this is owners may eliminate dogs from the breeding pool based upon a test, even if it's not relevant to their breed. Again, we're overachievers. So people will say, oh, I should not use this animal when that test is not appropriate for their breed. Furthermore, owners may get a false sense of health if the breed is clear for a number of tests. Remember I said there's 150 some odd tests? And people may say, great, I tested Fluffy, and they're clear of 100 different things. So is this a false sense of security? My dog is healthy. Or certain breeds will say, look, my breed doesn't have any of these conditions, so our breed is super healthy, we're done testing. And that's not a good perspective either. Other considerations is that the mutation is detected accurately, but it only accounts for a proportion of polygenic traits. An example is the alanine aminotransferase enzyme. A genetic variant that's associated with low normal. Is low normal bad? Maybe it's low normal is normal for that breed in blood chemistry panels. And that variant that's been tested accounts for 10 to 20% of the observed variation. So we've got a test, it accounts for 10 to 20%. What do you really do with that information? Many breeds carry that variant. So what does it mean for the breed if it's associated with low normal? And that's where breeders and owners need assistance in interpreting that information. Then there are multifactorial conditions. So dermatomyositis is seen in collies and Shelties, and this is work from Dr. Clark. And we typically hope for conditions that are governed by one gene so we can get a test for it. But most of the conditions are polygenic or multifactorial. And in this particular case, there are 3 different genes, the PAN2, the MAP3K, and the MHC, that interact to create risk. So if a dog has one of these 4 genotypes, they're at high risk for expressing this condition. These are associated with moderate risk. So 33 to 50% of dogs with DMS have this particular, one of these particular genotypes. And then we have low risks. So that's a lot of genotype. What does that mean? Well, it means that a dog who is high risk, bred to a normal dog or a dog with low risk, can have low risk puppies. So would you necessarily want to throw that dog out of the breeding pool? Not when we're thinking that it's important not to restrict the genetic makeup of different breeds, or to totally low risk can have a high risk as well as moderate risk. So it's very complicated, and that's the interpretation of the genes. Another aspect we have to think about are linkage-based, and they're not 100% accurate because recombination during formation of the egg and sperm can dissociate the marker from the trait. So we can separate the marker from actual expression, and those linkage tests are good, but not 100% accurate. So you have to be aware of that. Example, Italian Spinoni cerebellar ataxia, 95 to 98% accurate. Idiopathic epilepsy, there's a mutation on chromosome 37. Really low penetrance, low risk, and it's not even the actual mutation. So it just is a step toward. But you can get the results on a large panel assessment. Then there's genetic diversity testing, and that's a really hot topic because breeders are encouraged to maintain genetic diversity. But by definition, a breed has regions of homozygosity. So it's not necessarily, it's a work in progress. Understanding the value of genetic diversity in the testing realm is still under study. So implementing the tests, you need to know what the trait is it relevant, and is it inherited? You need to think about, if you're breeding, prioritizing those traits to be emphasized. Remember, even mixed breed dogs have a breeder behind them. Whether they're applying health testing or not, they have been bred. They don't spontaneously appear. And you also have to consider the population and breeding options for the future generations, not just this dog, but the future and the future of the future. And try not to recommend wholesale elimination of dogs, because you can genetically improve one trait but eliminate a lot of other traits that are very desirable. An individual is not a single trait. All of you in this room, I believe, carry 4 to 7 lethal mutations. You're not expressing them, but you certainly carry them, and you wouldn't necessarily eliminate you from the breeding pool. Are they essential? Yes, it's essential to do tests, all the tests. Is it essential to understand what they mean and to correctly interpret? Yes. Is it essential to record these tests? Yes. And this is, yes, I'm on the board of OFA, and the OFA is a repository for information. Owners should be encouraged to register their test results in an available repository, because other breeders can use that information and plan ahead, so that you can say, this dog doesn't express, but it has the propensity to pass on. I need to be aware of that so that I can make informed choices. And that's where you as veterinarians can assist. Testing is important for all dogs. Help in interpreting and not misapplying results. Use those results to generate healthcare plans. Help prospective owners look for healthy dogs that are produced by breeders who health test. As I said, mixed breed dogs have breeders too. It's just that they may not be paying attention to the particular kinds of genetic conditions behind their dog. So, thank you for your attention. And this little puppy says, I'm going to get health tested. How about you? And that is because every dog should be assessed for health tests and health screening. So thank you very much.